Hello guys, today's session at Medical Sciences Medicine by Naphtali Muhumuza. We want to discuss the anatomy and physiology of the ileum, which is the last part of the small intestine. In the previous video we have seen the duodenum, which is the first part. We said the second part is the jejunum. Jejunum comes from a Greek word jejun, which means dead. So we see nothing happening in the jejunum. So now we want to look at the anatomy and the physiology of the ileum. What happens in the ileum? And we know from the start we have been studying that in the ileum, which is the last part of the small intestine, that is where final digestion of food occurs. So we see this being the last part of the small intestine. And another thing, it is that is where final digestion takes place of food. And this ileum, it is folded into its blush borders, are folded into the villi, or what we call microvilli. It has multiple microvilli, and these microvilli are the one that increases the surface area. These microvilli are from the simple columnar epithelium. And this simple columnar epithelium having the villus, which are the absorptive cells, the villus. So microvilli are from the villus, and these are the absorptive cells. They are the one that absorbs food nutrients. Still in the video where we looked at the histology, we said it has also 4 layers, where he has the mucosa layer, having the epithelial, which is simple columnar, together with the lamina propria, which is alveolar tissue. Then we saw the muscle, we saw the submucosa, which is supplied by the Messina's plexus, or what we call the submucosa plexus. Then we saw the masticularis, made up of the outer longitudinal and the inner sacula. And this is made up of smooth muscles. And we said that in between the outer and the inner sacula, you have what you call mesenteric plexus. Then ends up with the serosa. So the mucosa layer, apart from having these absorptive cells which are calling the villus, it has other cells known as the Paneth cells. Paneth cells, it also has other cells called the crypts of Libacoon. It has other cells known as crypts of Libacoon. And these cells, the Paneth cells, the crypts of Libacoon, all of these are found in the enterocyte, or what we call the intestinal mucosa. And these cells, they are the one that collectively, they secrete succus entericus. Succus entericus, which we call intestinal juice. And this intestinal juice, we see production, apart from the intestinal juice, this juice contains compounds like the bicarbonate, which provide the pH, alkaline pH. It contains enzymes for final digestion, that is for final digestion of carbohydrates. So it contains enzymes, it contains electrolytes, not forgetting water, which carries 99.5%. So these enzymes, we have enzymes of which works on the carbohydrates, we have those which works on the proteins, and these of the carbohydrates, these of proteins. Carbohydrates, we have those that complete like maltase, we have sucrase, we have lactase, and dextrinase. Excuse me. And these are the ones that catalyze the final digestion of carbohydrates. Then we have the proteases. These proteases include dipeptidase. We find enzymes like aminopeptidase, which catalyze the final digestion of proteins. And we want to see the function of each. And we normally say that this enzyme catalyze hydrolysis reactions, whereby maltase, whenever we have maltose, this maltose is hydrolyzed by maltase enzyme in the presence of water to form 2 molecules of glucose plus glucose. Then we see if we have another substance like sucrose, maybe you can have lactose, you can have sucrose, we can have alpha limit dextrins. And these are acted on, these are hydrolysis reaction, they occur in the presence of water, where we see lactose is acted on by lactase, sucrose is acted on by sucrase, whereby it catalyzes its hydrolysis, and this one is dextrinase enzyme. And this one, the final product of lactose, is glucose plus galactose. This one is glucose plus fructose. And this one produces glucose. So these are the final digestion of carbohydrates in the intestines. Whereas for proteins, we see if we have dipeptidase works on the dipeptides to form amino acids. This is by dipeptidase. Then these amino acids which are formed, the aminopeptidase for them, now this dipeptidase, it does not work on dipeptides, but works on the tripeptides. Then this one, the aminopeptidase, the one that cleaves the final products of dipeptides to amino acid. This one forms dipeptide, not amino acid, this one forms dipeptide. Then these dipeptides which are formed are broken further to amino acids by aminopeptidase. Or in the presence of water, because these are hydrolysis reactions. And these are the ones that finalizes digestion of food nutrients ready to be absorbed either by active transport or facilitated diffusion. Those are all the major ways of absorption. So we have seen different cells being the last part of small intestine, final digestion, having absorptive cells, having Paneth cells producing mucus and the crypts of Libacoon. And we have seen the content which is in Succus entericus. Finally, we can conclude with the control. How are they controlled? And the control, still it is in 3 phases. We are looking at the regulation of intestinal juice production, phases of that trigger intestinal juice, what we have called the Succus entericus, and it includes the cephalic stage or phase, it has gastric phase, and it has intestinal phase. whereby the cephalic, it is always thought, smell, sight, when you see food, taste, you taste the food, that this triggers the autonomic nervous system and the enteric nervous system to release acetylcholine, which comes and stimulates production of secretions and movement, increase GIT motility. Then gastric phase, we see that presence of food in the stomach, presence of food chyme in the stomach, it triggers the intestinal cells like the Paneth cells and the crypts of Liebachon. There is also GIP, that is gastrointestinal peptide, which can go to inhibit production of gastrin because we no longer need gastric juice. But now, what we're interested in is the succus entericus. Then the intestinal phase. Intestinal phase is the last one, whereby presence of acidic food chyme. Acidic food chyme in the small intestine, we saw it triggering production of hormones like CCK hormone. We saw production of hormone like secretin. So production of GIP, that is gastrointestinal peptide. We can see production of bicarbonates and still also succus entericus. This presence of food, and we saw that this one, it does two roles, that is bile production and pancreatic juice. Then this one, these two, that is secretin, GIP, and somatostatin. I can also add somatostatin. They inhibit production of gastrin. That is when they are produced. Then also they stimulate production of intestinal juice. This control is both, if I say control of this small intestine secretion, it is both duodenum and ileum. And that is how these are the phases which it can be controlled. That is cephalic, gastric, and intestinal phase. Thank you so much for always listening until the end. Always put your comments, like the video, and share. Thank you so much.